Hello, everyone. My name is Rael. I have Samuel and John here with me today. And we're going to be talking a little bit about RTK PI3K activation and how it's used as an escape mechanism for the typical MAP kinase pathway. So let's get right into it. So first thing I want to talk about is what we actually know about colorectal cancer, which is our specific target cell that we're going to be talking about, or cell line. So first of all, it's the third most prevalent cancer worldwide. So it's a really prominent issue. And the next thing I want to talk about is it's focused on the EGFR growth factors, or epidermal growth factor receptors, right? So what this means is it affects the signal cascade so it can't operate in its usual manner, which allows for tumor growth and reduced suppression of that growth. Two vertical suppressing treatments that are usually used is cetuximab and rifaxitinib, or we just call them CTAX and Reframe usually, and they've generally pretty good. However, with the KRAS mutation, these methods can have a reduced effect or even no effect at all. And what this paper is trying to do is figure out what is the mechanism of that escape. So I'm going to show you a figure in just a moment, but before I do that, I want you to consider a few things. So first, the MAP kinase cascade is autophosphorylated by the EGFR, which is what we already talked about. And two, the IP3 pathway is very similar to the MAP kinase, which also starts with phosphorylation, but the PI3K offers an alternative route to growth factors. So this helps the mutant cells survive when they shouldn't be allowed to with the treatment that we're providing. So as we can clearly see right here, this is in red. This is the typical MAP kinase pathway that the cell is going to do if it has the resources it's allowed. However, if we block that off with our two treatments, it's going to take this PI3K pathway instead. And what our study is trying to do is figure out that exact mechanism. So immediate research that needs to be done in this area is, like I've already said, find the AKT pathway and the MAP kinase pathway activity, how the escape mechanism is happening, and how the KRAS mutation is related to those, as well as finding a drug combination that is more effective and allows for greater cell death for those mutated tumor cells. I'm going to pass it off to Samuel now.
All right, so I'm just going to get a bit into the methodology and some of the brief results of our study here. So the four cell lines that were analyzed in the study were HCT15, HCT116, SW480, and the LOBO cell lines. Initially, they were cultured for 6 to 9 months with increasing concentrations of the anti-EGFR antibodies, which was cetuximab, and the MEK inhibitor, which was rifaxitinib. And it was observed that the IC50, or the inhibitory concentration at which 50% of the cells become inhibited, for the four cell lines increased between 17 and 190 fold. Furthermore, they then did an immunoblot, or a Western blot, analysis of the proteins and their phosphorylation in the cell lines, their parent and the resistant cell lines. And so they were looking at the PI3K and AKT pathway proteins. And what they observed was these pathways were being active, as evident by their effectors being active, which were S6RP and 4EBP1. They were being actively phosphorylated. So then now with an establishment of active PI3K and AKT pathways, they looked into the theory that this activation could be by various receptor tyrosine kinases. So to start, they looked into the expression of various RTKs, and their qPCR determined a greater expression of the following RTKs: EGFR, which was currently being suppressed by the antibodies, the IGF1R, and the HER2 and 3 RTKs. And then following an immunoblot, they did observe greater phosphorylation in all of the above RTKs. So with greater phosphorylation of the RTKs and the activity of those two pathways, the question then became, are these events connected? So they performed a knockdown of the RTKs and observed that the PI3K and the AKT pathways were significantly less active with IGF1R and HER2 knockdown, suggesting that those receptors do activate those pathways. They also looked into the activation of those RTKs using a microarray analysis, looking at ligand expression for those RTKs. And they did observe in some of the cell lines an increased expression of some of those ligands, such as TGF-alpha, epiregulin, and amphiregulin.
I'll be talking a little bit of the results, analysis, and the conclusion here. So the four main kind of results and analysis that we saw in the paper was, the first is They identified the human CRAS mutant CRC cell lines that have a resistance to both the combined treatments of cetuximab and rifampicin. So those were the four cell lines that were mentioned above in the methodology. And yeah, so the second part is that they examined the emergence of the AKT pathway activation with the vertical suppression of EGFR and MEK in those CRAS mutant CRC cell lines. So in those cell lines that they first identified, they then looked at the AKT pathway activation with the vertical suppression alongside it. Noticeably, this experiment demonstrated both that PIC, a PI3K alpha inhibitor, can also restore sensitivity of the CRC cell lines with cetuximab and rifampicin. This was shown with the true Talile method, which both validates this section of the results, but also warrants a little bit of criticism that we'll mention later. Third point that we have is we also investigated the role of RTKs in the transcriptional adaptation and upregulation in those CRC cell lines. So we used a Western blot and a baseline microarray gene expression, which were used to identify the pathways related to the resistant phenotype. So looking at that resistant phenotype in those CRC cell lines. And finally, the researchers examined the mechanisms of activation in of the RTKs in therapy-resistant cell lines. So they used microarray analysis and immunoblotting to see that the ligands of several of these RTKs were also upregulated and increased in activity. So in conclusion, the researchers claim that transcriptional adaptation is the reason for the PI3K activation with vertical suppression of the EGFR and MAPK pathway. This is done through the upregulation and cooperative activation of different RTKs within CRAS mutant colorectal cancers cell lines. A minor criticism that we found is that they do not mention the fact that the combination of the PIC and the cetuximab and rifampicin can restore sensitivity, as mentioned before, of the original treatments. And they never mentioned or talked about that, which we found a little bit surprising. And the claims that were proposed in the conclusions were actually validated. So the ones that they did say through the experiments conducted and the results found. And this can initiate a branch of research, which could aid the medical field greatly, especially in the colorectal cancer research area. And with that, here's a little bit of the references, and thank you for listening.